Hello all, thank you very much for being here. Today I would like to present my summary research about the role of the IL-27 signaling pathway in the regulations of regulatory T-cell survival and metabolisms. My research focuses on the unknown underlying mechanisms of graft versus host disease. So what is graft versus host disease? Graft versus host disease, or GVHD, is a complex multisystemic disease usually following hematopoietic stem cell transplantation. GVHD happens when donor T cells recognizes recipient tissues as foreign and starts to attack them. This leads to systemic inflammation, with increased production of inflammatory cytokines and activated alloreactive donor T cells. Without a regulatory mechanism to control this inflammation process, it has been shown that regulatory T cells or Treg cells can suppress the severity of GVHD. Therefore, establishing a more effective network of this T cell population has become the potential treatment for acute and chronic GVHD. Previous data from Dr. Drobyski lab has shown that blocking of IL-27 signaling, a member of the IL-6 cytokines family, can reduce GVHD severity and increase the reconstitutions of Treg cells by prolonging their survival. Therefore, our hypothesis is that IL-27 plays an important role in regulating the metabolic and anti-apoptotic pathways that can prolong the survival of CD4 plus regulatory T cells. To test this hypothesis, we harvested the spleens from wild-type mice, which will act as our control, and IL-27 receptor knockdown mice. Treg cells were isolated and put through flow cytometry for sorting. Figure 1 is our cell sorting data, which shows high purity of Treg cells population, 99.2% in wild-type sample and 98.9% in IL-27 receptor knockdown sample. The sorted Treg cells will be used for two different analyses. One is Western blot and another is for cell cultures. Although our result on Western blot did not show any statistical difference in the absolute expression levels of pro-apoptotic proteins, including Beam back and anti-apoptotic proteins, which are BCL2 and MCL1, when we compare the relativities of expression levels between each pair of anti-apoptotic and pro-apoptotic proteins, there was a significant elevation of BCL2 beam expression ratio in Treg cells lacking IL-27 receptors. This means that IL-27 receptor deficiency, Treg cells are less sensitive to apoptosis. To explore further the effect of IL-27 on cell metabolism, we harvested the remaining sorted Treg cells that had been cultured for additional 36 hours in medium containing anti-CD3 antibody, anti-CD28 antibody, and IL-2. We use seahorse technology, which will target components of electron transport chain and create certain cellular stresses. This technology will measure the mitochondrial respiration rate by oxygen consumption rate, or OCR, and to assess cellular glycolysis by quantifying extracellular acidification rate, or ECAR, under stressful situations. Here we could see that in cells lacking IL-27 receptors, which is shown in the blue color, both basal and maximal mitochondrial respiration rate was significantly elevated, with increased ATP productions and larger spare respiratory capacities. In addition, our ECAR analysis demonstrates that in IL-27 receptor deficiency Treg cells, shown as red color here, glycolysis was remarkably increased with larger glycolytic capacity and reserve. In conclusion, Although there was no statistical difference in the absolute expression levels of pro-apoptotic and anti-apoptotic proteins, there was a significant elevation in the BCL2 and Beam expression ratio in Treg cells lacking expressions of IL-27 receptor. This indicates that IL-27 receptor deficient Treg cells are less sensitive to apoptosis. Additionally, our seahorse analysis also shows that the absence of IL-27 receptor appears to alter metabolic function of cells under stress, with increased mitochondrial respirations and glycolytic rate to promote the survival state of cells. This suggests a critical role for the IL-27 signaling pathway in maintaining the metabolic fitness of CD4 plus Treg cells. With that conclusions, in the future, we will further confirm the role of IL-27 in the regulations of Treg cells by generating induced Treg cells with different concentrations of exogenous IL-27 in vitro. And we also will examine the AKT pathway, which plays an important role in the regulation of apoptosis and metabolism in IL-27 receptor deficient Treg cells, to investigate how IL-27 regulates Treg cells function. Last but not least, I would like to thank the Center for Immunology for their financial support, the MCW core laboratories, instruments, and facilities, and the awesome mentorships of Dr. Drobyski laboratories members. Without these people, this project would not happen. If anyone has any questions, please feel free to reach out to me. Again, thank you very much for your time and your attention.